One of the useful applications of genetic engineering is something called in vitro cloning. And this is a technique that basically combines several of the techniques that we have been talking about in Chapter 10. In the in vitro cloning procedure, what we want to do is take a gene that we're interested in studying or a gene that has some use to us, and we want to copy that gene and place it into a bacterial plasmid and get a bacterial cell to make the product of that gene for us so that we can either use that product or study how that gene works. And for this example, I'm going to use the human insulin gene. We do use bacteria to make a number of human pharmaceuticals. So they can be used to make insulin, a variety of human hormones like human growth hormones or hormones for hormone replacement therapy. They can be used to make amino acids, vitamins, a number of different things that humans need. We can actually get by turning bacteria into little factories for us to make these things. So we can induce bacteria to make insulin, and first we would have to isolate and purify DNA from a human who has a functioning insulin gene. Then we would use PCR to make millions of copies of that insulin gene. Someone has already done the sequencing, so they know where to place the primer so that we get a good copy of the insulin gene. Now we have millions and millions of copies of that DNA to work with. And once we've done that, we use our restriction endonucleases, our enzymes, to cut on either side of that insulin gene so that we get these nice sticky ends. We want sticky ends when we are trying to do cloning because this sticky end is going to be complementary to the sticky end of our cloning vector. So now our target gene is ready to go. Now we need our cloning plasmid. So we actually harvest plasmids from bacterial cells and modify them, and we can use these plasmids for a variety of things. And one of the things we do is to splice other genes into them. So we want to prep our bacterial plasmid by digesting it with the same enzyme that we use to trim the ends off of our insulin gene. This is going to open up the plasmid, and now the sticky ends are going to be complementary to the sticky ends of our insulin DNA fragment. So these little bands are going to match up. So here we're only going to cut once, opening up this circular plasmid so that we have these sticky ends. And now we're going to mix our fragments of DNA with our human insulin gene into a test tube with the digested cloning vector, and the sticky ends are going to line up. They're going to find each other, and that's why we wanted to cut them with the same enzyme to get these nice sticky ends. So they start to go together all by themselves. And we will then add an enzyme called ligase that is going to create the bonds between those two DNA molecules. So now we have the human insulin gene spliced into a bacterial plasmid, which is known as the cloning vector. Now we use the process of transformation. We know that some bacterial cells are naturally competent. In this case, we're going to make them artificially competent and induce them to take in these plasmids. And these bacterial cells, because they have taken in a plasmid which contains a human gene, are now going to make that human protein. In this case, we put the insulin gene in. The protein is insulin. They're going to make insulin. They don't use insulin, so the insulin is secreted into the bacterial media, and now that insulin can be cleaned up and purified. And this is a relatively routine procedure in this day and age, and we can induce bacteria to make any number of products for us. We can actually now take this further and inject these genes via transformation and transfection into eukaryotic organisms. And that's how we get things like cats that glow in the dark because they have jellyfish DNA and other weird things like that.